My name is Jim Polli from University of Maryland, and I'd like to talk to you about a workshop summary report that's being published in 2012. This workshop summary report concerns a workshop that was held in late 2011, an AAPS workshop regarding facilitating oral product development and reducing regulatory burden through novel approaches to assess bioavailability and bioequivalence. Lots of people, academics and industry contributed to this workshop. And in particular, special thanks to Jack Cook, Barbara Davitt, and Paul Dickinson. Okay, there's really two main elements of this workshop, again, which is summarized in this workshop summary report published in the AAPS Journal. The two areas were best practices in oral drug product development as well as evolving bioequivalence approaches. With regard to best practices in oral product development, this workshop was really an opportunity for practitioners to get together and talk about many things, including identifying development challenges towards successful oral drug product development. As you know, it's difficult to develop drugs, difficult to develop drug products, including oral products, and this was an opportunity to really focus on what are the challenges and potential solutions facing the industry with regard to developing things like tablets and capsules and things of that sort. Another discussion point was the integration of biopharmaceutical data with other drug formulation and patient disease data. The way in which various companies and practitioners approach this are actually quite different, and it was an opportunity to sort of compare notes about how different companies do development. Hot topic, as you know, is QBD, or quality by design, and what are these various approaches to performing so-called quality by design and in general approaches towards drug product developability paradigms. There's also a fair amount of discussion about what tools are available in early development. Things like critical absorption factors, how can they be assessed and incorporated into various oral absorption model building in order to assess whether a potential product has high risk or low risk. You may have heard of QTPP, Quality Target Product Profile, as well as root cause and risk analysis as tools to assess product failure. These were also discussion points. A lot of discussion about, what is it, how can we go about reducing risk and meet timelines, since timelines in the industry are getting tighter and tighter with fewer and fewer resources. Also, the aspects of the biopharmaceutical classification system, as well as in vitro and vivo correlations. What are their current state of the art in facilitating product development, as well as post-approval issues. Okay. So in addition to best practices in terms of oral product development, a second area of focus of this workshop was evolving bioequivalence approaches. Two big areas that were discussed were highly variable products as well as specialized modified release products. With regard to highly variable products, there were speakers from all over the globe. For example, in the U.S., there was discussion about the reference scaling average bioequivalence approach, as well as the two-stage group sequence design. Canada is now indicated an acceptable approach includes adaptive sequential design. And in EU, two-stage design and ongoing efforts to consider widening Cmax acceptance limits are still of current thought. A second topic involving bioequivalence were specialized modified release products. The FDA has discussed quite a bit about partial AUC for certain products, products that where they're modified release, but there's certain elements of the early and maybe middle as well as late elements of the product profile. Discussing rationales and limitations of certain metrics were discussed. What aspects of partial AUC for various products could be considered. A big ongoing theme in the era of bioequivalence continues to be the benefit of needing community development, validation, harmonization of newer bioequivalence methods, including BCS biowaivers, as well as for highly variable drug product trial design. Okay. Future improvements was a big discussion point in both of these areas, best practices as well as evolving bioequivalence approaches. With regard to best practices in development, modeling to establish critical product attributes was a big discussion point. The more modeling that can be done earlier on in development has the promise to reduce risk and make, the idea of fail fast continues to be of interest. Okay, so ongoing elements include how to implement quality by design. Quality by design has been with us for several years, and various companies have had the opportunity to implement it. Others look forward to implementing it in the near future, and it was an opportunity to assess how is that going. Again, with the goal of improving clinical efficacy and safety and reduce patient variability in order to really facilitate development and really just do better science. There was also significant interest really in modeling and doing a better job of that early on. And it was concluded there's really a need for collaboration, since no one company or institute or a group can really do this alone given the challenges. So there's really a need to validate in vitro models that are required. Just some topics include supersaturation, gastrointestinal motility, regional absorptions within the gastrointestinal tract. All these in and of themselves are pretty complex, so there's a need for us to work together. Okay. It's also recognized that current models in terms of oral absorption, including predictive models, really rely on a limited number of permeability database, and there's a need to expand knowledge and really to get increasingly better tools to do this sort of thing. Okay. With regard to bioequivalence approaches, it was highlighted that there's a lack of consistency of acceptable regulatory practices across the regions around the globe. For example, allowing scaling in the analysis of BE trials, bioequivalence trials versus not allowing dissolution-based biowaivers for BCS class 3 drugs, as the case for Europe, versus none for the US currently, versus no BCS biowaivers at all, as is the case of Japan. So this lack of harmonization leads to a number of different things. Global companies are forced to resort to bioequivalence methods that are acceptable to all regulatory bodies. These often turn out to be really the most resource intensive and time consuming. And there's also this element that this sort of approach where we always do in vivo studies really is perhaps broadly not the most ethical approach. Okay, so there's a need for the scientific and regulatory community to harmonize newer BE approaches, including BCS biowaivers as well as in vitro trial design. Okay, so in this workshop report, to which many people through the industry contributed to organizing the paper as well as the workshop itself, which it was in late 2011. That's actually been rolled into a broader AAPS Journal themed issue involving oral drug product development and performance. A number of different ones involve broadly looking at various aspects of oral product design and development. A number of others looked at BE in particular, and still others looked at technique issues and technical issues with regard to various aspects that come up in developing oral products, various oral products. So the intent here is to really have a themed issue in the AAPS Journal that will be valuable to practitioners in the area of developing oral products, and many of these are new publications, and additionally the workshop report will summarize many things with regard to the various talks that were given at that workshop. Thank you.